Hey, I'm back, and it's the Hemoglobin Challenge, episode number 5. Yep, we need a 5th episode because ICR has done it again. They're talking hemoglobin in fossils. They're talking about red blood vessels, red blood cells, and they're even talking about finding chlorophyll in fossil fuels. What's up with that? Let's call out ICR for inappropriate use of biomolecular terminology and fossils. And in order to do that this time, first I'm going to show you a transcript of a recent video that they made, and we'll look at the language they've used. Then I'm going to go to a 2022 review article on the biochemistry of biomolecules found in dinosaur bones. And we'll take a look at the types of molecules that have been found in dinosaur bones and look at why some of those particular molecules are found and not others. So let's get after all that coming up. Yeah, I'm a little surprised that I have to be talking about this again and challenging young Earth creationists to be more careful with the terminology they use when they refer to biomolecules being preserved in the fossil record. The hemoglobin challenge is really about primarily about the use of the word hemoglobin, but it's also about lots of other different biomolecules that are found in the fossil record. Hemoglobin in particular, or hemoglobin is a large, complex protein molecule that's found in embedded in the plasma membrane of your blood cells. What is sometimes found in fossils are the decayed remains or evidence of the prior existence of hemoglobin in that particular location. And in fact, it is some of the actual atoms that existed in that hemoglobin molecule, but it's no longer hemoglobin. It is a very stable byproduct of a breakdown process called heme. And these heme units are found, and they do indicate, as I said before, that hemoglobin was present at that site. But it's improper to say that hemoglobin was found inside of dinosaur bones. Right? It gives the impression to the audience that fresh blood was found there, that something that hasn't decayed at all is remaining still in these fossilized remains of dinosaurs. Right, that's the big problem. So what I'm going to do is I want to show you I'm just going to show you a transcript of a video where there was a long discussion about this at Institute for Creation Research yesterday. I'll show you their use of the terms, and then we'll talk about a better use of those particular terms. But I also want to show you, as I said in my intro, I want to show you a paper, and I want to look at some of those molecules themselves. So that's something I'm adding into this particular episode number 5 that you haven't seen necessarily if you've watched the other prior episodes of my hemoglobin challenge. All right, let's get to it. So here we are at the Institutes of Creation Research. They did a live video in which both Dr. Thomas and Dr. Clary, all right, two members of their team, were talking about soft tissues, and specifically, do fossil bones have blood vessels? But of course, that ranged into a whole bunch of different topics. So that's the video I'm referring to, and I'll put a link to that at the bottom of this video. All right, so I just downloaded the transcript off of YouTube, scanned through there, and I listened to it. So I knew that they had talked about this. And I will highlight a few things here. So 15 minutes into it, we see what was actually done was you see that there's these proteins and structures and blood vessels and things in these fossils. Right? And they just can't be there because if they're that old, right, how could they possibly be there? And besides, you can't verify the ages, because we don't trust radiometric dating and so forth. All right. Lots of other content in this particular video that we're not interested in at this particular moment. So once again, I got to remember where this was. Here we go. At the 21-minute stage, we have, to me, the interesting fossils are the ones, like when you are looking at oil and you put that oil on a microscope and you can see chlorophyll, right? Remember, this is a transcript that YouTube has made, so sometimes it's not all the best in terms of the interpretation, but I do remember hearing him say, if you put on a microscope some oil, all right, fossil fuel oil, you can see chlorophyll. I mean, you can't see it in the microscope, but you could do tests for that chlorophyll. Yeah, right. He's correcting himself. You can't actually see that molecule, but you can test for chlorophyll and find out there's chlorophyll there. And of course, that should have biodegraded long ago, right? Because oil is supposed to be millions and millions of years old, hundreds of millions of years old. And chlorophyll is this fairly delicate molecule. Yes, I mean, what happens in the fall, right? The leaves change color. Why do they change color? Because the chlorophyll is no longer being maintained, because it takes a lot of energy to constantly remake chlorophyll and maintain chlorophyll in plant cells. Once the chlorophyll begins to degrade, it reveals other more stable pigments, and those reflect different colors, and therefore the colors of the leaves change. There's your little extra bonus botany lesson for the day. All right. But anyway, the idea is that chlorophyll is a very unstable molecule, and it does begin to fall apart, begins to degrade. Right? And so he's right. Well, why would there be chlorophyll present? And yet he's claiming that there's chlorophyll present in oil. This actually is not true. You cannot find chlorophyll in fossil fuels. What you do find, though, is you find an extremely stable endpoint or a breakdown product of chlorophyll. So chlorophyll breaks down into, and I'll show you the molecule in a few minutes when we look at this other paper on different molecules that are found in dinosaurs. It breaks down into a much smaller molecule, of which is very, very stable, and that is found in oil, and that's been known to have existed for a long period of time. But it's improper to say that chlorophyll is found, because once again, he's playing on the fact that at least some of his audience understands that chlorophyll is an unstable molecule, and therefore can't exist in oil. And that seems to make the case for a young Earth, right? That chlorophyll exists in this oil. You're getting also down here, so you in highlighted here, you're getting collagen protein sequence. And we know that that's what blood vessels and skin are made of. And collagen is also embedded in bone tissue itself. And so in some of these samples, it looks really freshly deposited, and it really does not, and it does fit thousands, but not millions of years, right? So there's collagen, right, that's been found in these dinosaur bones, and collagen's found in blood vessels and skin and inside bones, and it kind of looks like that must be fresh, because if there's collagen there, it must be freshly laid down, because collagen also should break down, and we know that under certain conditions it does break down. Let's scroll down here to the good stuff. Leads me to say we've talked about blood vessels. We've talked about collagen soft tissue. It's kind of a broad topic, or at least it seems like a broad topic. What kinds of things that we're seeing other than blood vessels and collagen? Okay, blood vessels in collagen, and I'll let you extend the list first. All right, I think this was Brian Thomas talking to Clary about this. And if you have anything to add to that, I'll extend it to your extended list. Now, Dr. Thomas has been keeping a list of all the publications out there that includes something about soft tissues. And he has that available for anyone to be able to see. I don't know if you know enough about protein name that I talked about in blood cells. Some of their red blood cells aren't really that. They double as red blood cell-like things. Now, despite the translation here, what's really being said here is he is trying to say, not very clearly, but he's trying to say that the red blood cells that have been said to have been found in dinosaur bones, they look like red blood cells. They're not actually like a living red blood cell or a fresh red blood cell. I don't think his audience really understood what he's saying. I understood what he's saying, and I'm going to congratulate him for that, because that's an important thing for him to say, is that those red blood cells are things that look like red blood cells, right, in those fossils. They aren't actually red blood cells. Same with the blood vessels. If collagen fibers that were present in, or elastin fibers that are present in blood vessel walls are preserved, the actual cells that make up the blood vessel walls aren't there. All right, so there is some biomolecular molecules that are preserved and represent actual remains of biomolecules from the dinosaur tissue itself. But the entirety of the cells, all right, don't exist anymore. And that's what leaves the outline of blood vessels. So you can talk about blood vessels, but you need to be able to say there are the broken down remains of blood vessels are present. But let's not get distracted on the blood vessel thing. That's a harder thing to talk about. Let's go right to the hemoglobin part, because that's what I'm interested in. Let's scroll down here. Some of these proteins, but we're finding, like you said, hemoglobin. They've published elastin, laminin, ovalbumin, different proteins than what they're specific and what they're using specific chemistry tests to find. All right, and that's we're going to look at this other paper at those types of chemistry tests. All right, so here he's actually listing a number of different molecules that are found in the remains of dinosaur bones. And the first thing he's saying is you find hemoglobin. All right, so that's our big word, right? Hemoglobin. But he's also mentioning you find elastin, laminin, ovalbumin, so I have to show what those are as well. But he's saying they found hemoglobin, and that's what I'm saying is an incorrect use, right? It's describing the biomolecule that was found incorrectly. They can say that evidence of the presence of hemoglobin was found. There is a breakdown product of hemoglobin that's still present in those dinosaur bones, but it's not actually hemoglobin, because hemoglobin consists of a certain biomolecular structure. That certain biomolecular structure that makes hemoglobin is not present in those bones. Now, throughout this, they're going to mention hemoglobin, blood cells, blood vessels, collagen, multiple different times, pretty much repeating the same thing over and over again. Let me remind you again why I get a little testy about the use of the word hemoglobin and the use of some other biomolecular names. Here's my analogy. If I said I found $1,000, if I told you I found $1,000 bill digging in my backyard, all right, I found this fossil, right? It's $1,000 bill. And you're like, well, let's go out and spend it, right? You would think that I'm talking about $1,000 bill because that's how I described it. I said $1,000 bill, and that's going to conjure up an image in your mind. But what that $1,000 bill was is just a little tiny fragment of it, just enough that I can see and identify that that's what it was. It was $1,000 bill, but if I only have this tiny little fragment of $1,000 bill, I don't actually have a $1,000 bill in my possession, do I? I have the remains of a piece of that $1,000 bill. And saying you have found hemoglobin is the same thing. You're saying you found hemoglobin, and I'm thinking, oh, this really complex protein. You found that? But no, that's not actually what they mean. They didn't find hemoglobin. They found a small portion of the hemoglobin molecule, which is just the heme unit, which is this ultra-stable sort of central core. And that is enough to say, because of the iron present and the molecular configuration of that heme molecule, to be able to infer that at one time was part of a hemoglobin molecule. And so you'd say, like, yes, we found the presence of past hemoglobin at this particular location, but we didn't actually find hemoglobin. Found the remains of a piece of hemoglobin, just like I found the remains of a piece of a $1,000 bill. Now, that's not nearly as impressive if you're trying to convince somebody that biomolecules haven't broken down and that's why the Earth is young. Because how could they possibly have found hemoglobin when it breaks down? Well, they didn't find hemoglobin. Just like, how could you find a crisp, brand new $1,000 bill underneath the ground if it had been there for the last 50 years? And it's like, nope, I didn't actually find a crisp $1,000 bill there, right? I said I found a $1,000 bill. I found evidence that there had been a $1,000 bill there, but it had decayed, leaving me just enough to know that it had existed at that point. All right, here's the paper I wanted to show you. Chemistry and Analysis of Organic Compounds in Dinosaurs. This is from 2022. So this is a fairly recent sort of review of all the different types of biomolecules that have been found there. And I'll just read you the simple summary here. Fossils of dinosaurs, other than birds, are at least 66 million years old. Nonetheless, many organic compounds have survived fossilization, right? I agree. And I think most scientists agree. There are many organic compounds that survive fossilization and can still be found in the fossils. This article describes the discovery of organic molecules in dinosaur fossils. It provides a review of the analytic methods used for their detection and characterization and presents the wide range of chemical or organic compounds, including small molecules and polymers. that have been found in dinosaurs to date. The difficulties in unambiguously confirming the presence of some of these organic molecules in these fossils are also discussed. So how hard is it to actually identify whether it came from the actual dinosaur itself versus some kind of contaminant, and just the difficulty of some of these methods for being definitive about the actual molecular bonds that are found and the identification of the molecules themselves. All right, there's a long history of finding organic molecules and the first identifications of them. Of course, Mary Schweitzer and other things are identified in here. And then it talks about the different methods that are being used. All right, so spectrophotometry, right? There's infrared and Raman spectrophotometry. We've got mass spectrometry. So we have a variety of forms of spectrophotometry. We also have immunological techniques. Well, immunological techniques are a method of, essentially you can think of that as staining, right? Or binding something that you know will only bind to certain other types of molecules. And if you find there is binding that occurs, you can infer that that other molecule is present. So there are multiple methods for identifying molecules. Most of these require quite a few, I'll say, specialized skills to be able to do that, along with usually very expensive equipment. This is interesting because ICR at the end of their discussion talk about how they're setting up a lab to be able to study biomolecules and biomolecular preservation. And that's really expensive work if what they're talking about is, mass spec and things like that. And I don't think that's what they're talking about. But that's the kind of thing they really are going to need to do if they want to truly get down to the intimate details of the molecular structure of these molecules. All right, so what kind of organic compounds are found in dinosaurs? And what's, I think, an important overview here is that there are certain classes of molecules that are found in dinosaur remains and other fossils that are hundreds of millions of years old. It's not just any and every possible biomolecule. It's very specific types of biomolecules, and generally we consider these biomolecules to be highly stable polymers generally, or cyclical aromatic compounds that don't degrade easily under a number of different conditions. That's the general theme here. So biomolecules most certainly are found, but they're of certain classes of molecules, and they're generally modified from their original state. Their original state meaning the state they were in when they were in a living thing. So for example, there are a variety of different forms of pigments. That would include eumelin and felaminin. I can never say that word. A variety of different carotenoids and so forth. And I will show you a little bit, just a tiny taste of the biomolecular structure of those to show how stable they are. There's porphyrins, which also include some of the same molecules. And again, porphyrins are breakdown molecules of things like hemoglobin or of chlorophyll. So here we go. Here's heme. Now, this is the one I've been making a big deal about, right? The heme structure is this central aromatic cyclic compound. It's carbon organic compound with this iron in the center. And it is found in a number of different bones from dinosaurs, right? And so we would say that that is evidence for the fact that there was blood present there at one time, but it actually isn't the hemoglobin itself. Oh, and then in eggshells, there are some other similar compounds that have similar structures to them. Just scrolling through here, these are pheomelin and eumelin, which are the molecules that are found in, say, squid ink, that are found in the melanocytes, in feathers and in the skin that reflect certain colors and so therefore are part of the coloration of skin and feather types. And again, these are cyclical, double-bonded organic molecules that are considered to be quite stable. And it's not even that this exact molecular form is found in these fossils, but rather it is sort of the core of these fossils that has the core of these molecules, enough of the molecule with enough bonds in certain positions for us to be able to say, oh, that is or was that particular molecule in the past. But it's been highly modified over long periods of time. I'll put a link to this particular paper. It's open access, and I encourage you to look at that and read about these different compounds if you're interested. Collagen is a polymer in the sense that it's a repeating unit. It's a repeating unit of amino acids, so it is a type of protein, but it's an extremely compact protein of the same exact amino acids over and over and over again. And then it forms these long protein fibers, but then these protein fibers are stacked on top of each other, all right, in close proximity, and then they end up binding to each other, all right? So a variety of different form of bonds, including even covalent bonds at some point, begin to form, which bind all this together, and that's what makes collagen so strong. And the thing about collagen and the particular amino acids that are present there is that it is water, it's hydrophobic, right? So it resists water. Therefore, water is one of the biggest things that's going to damage, or it's going to cause hydrolysis and damage molecular bonds in these various proteins. And so being in the center of bone, and then having collagen there, and then having it resistant to water, and then having it preserved, encapsulated in bone, means it's not surprising that you're going to find collagen, the remains of collagen fibers, still present. Even so, even collagen is been modified, all right? Its molecular bonds are somewhat modified, but there's enough of the same bonds that are these repetitive polymer bonds present there that we can identify this as collagen fibers. And this is probably what's being found in terms of these organic molecules that make up things like the things that look like blood vessels. Right? They are the remains of actual pieces of blood vessels that are there, but there are these extremely stable remains. All right? Collagen being very, very stable over periods of time. Now, and ICR is correct. Under certain conditions, collagen will decay. But in the conditions found in these dinosaur bones, there isn't any reason to believe that it has to have decayed. It's not shocking that they would find these collagen fibers. All right. These tables just tell you where they've been found in different bones, but also the different methods used to identify the collagen. And then as we keep going down here, we have keratins, all right, which are found in feathers, and they're also very similar, right? They end up being a polymer. It's a repetitive unit of the same amino acids again. Again, those amino acids sometimes have had their side chains modified over time. And so you're not going to find the exact molecular structure that was present there in the original keratin. But because the keratin forms these sheets, right, and these sheets stack up on top of each other, and then the sheets themselves actually have secondary binding of the different layers together, all right, it's not going to be just the original amino acid string. Which if it was just by itself an amino acid string, yes, that would have very likely have decayed long ago. But then when you stack these on top of each other and you bind them together, you end up with a much more resistant, much more stable organic compound. So you either have these cyclical, right, aromatic, double bond, high frequency of double bond carbon organic molecules, which are really the core of what were more complicated molecules in the past, but the side chains have been removed, and you just end up with this core that's very, very stable, right? So you either find those types of things, the heme units, or you're finding these long stretches of polymers in which the polymers are braided together, stacked on top of each other, bound to each other as well, and typically hydrophobic. All right, so resisting having any kind of water come into and mess around with the bonds. This review provides a collection of organic compounds identified in dinosaur bones and soft tissue to date, giving insights into their chemistry and analytic techniques used for their identification. Reports on organic compounds are increasing as more targeted, sensitive analytic approaches that use less and less sample material are being developed. Organic compounds detected from dinos so far comprise pigments such as porphyrins and melanins, and proteins including collagen type 1, collagen type 2, and keratin. If these were organisms that had just died recently, it would be easy for these techniques to identify lots of different things and to be able to sequence actual different amino acid sequences from different proteins. And we'd have an abundance of different kinds of evidence for that. Just look at, let's put it this way. If you go back 4,000 years, 4,500 years, right? That's the time of the flood. They say that all these things are only 4,500 years old. I'm sure Genesis and and ICR should be able to find samples that they think are more than 4,000 years old, right? That have been preserved under really awful conditions, like awful meaning they should have led to a lot of decay. And I would say they're still going to find lots of organic remains, lots of organic molecules, all kinds of different organic molecules. Woolly mammoths, right? We have ones that are thousands of years old, and they actually do have red blood cells, like actual cells. Remember, if there were blood vessels and those blood vessels were actual remains of cells, not just fibers that were in cells, if there were actually cells there, it would be very simple to show that they had cellular structure, right? You do some kind of, you do scanning electron microscopy. You would freeze it and you would shatter it and you would show the different organelles that are present there. That would be relatively simple stuff. Even ICR should have the technology or be able to hire the technology to do that, to get that kind of evidence. And I don't know why any other scientist hasn't done that, because that would be low-hanging fruit. They're going around doing these really complicated, highly sophisticated types of studies in order to eke out some little bits of data that suggest there were biomolecules there. Versus, hey, if there were red blood cells there, I should be able to just do an extraction and be able to extract a number of different biomolecules through just basic organic chemistry, right? I should be able to take the phospholipids, which represent the phospholipid bilayer of a cell, right? I should be able to just extract those easily and then be able to do some fairly simple analysis to identify that I have phospholipids. There's a whole bunch of other proteins there. I should be able to pull out a bunch of proteins. I should be able to run them on a gel. I'd be able to do some kind of ELISA or some kind of, well, I guess. Anyway, this paper, I didn't finish reading the abstract. Chemical imaging on sample surface using time-of-flight secondary ionized microscopy and other more invasive techniques, namely liquid chromatography, coupled with tandem mass spectrometry, were also employed. Yet, even as analysis techniques become more advanced and highly sensitive, it still remains challenging to prove the endogeneity, in other words, that these are molecules that are found that are dinosaur remains, not just contaminants from the environment, of the detected spectrum, especially when searching for proteins or DNA. Further development of sample preparation techniques that minimize contamination is required. So there's still a big concern about contamination. Some of these techniques are super sensitive and you're using small amounts of sample. And so if you had a very, very small bit of contamination from, anything, if I have a little tiny bit of my skin, all right, it's going to show a whole bunch of different proteins being present, but those actually aren't present in the dinosaur. And so you need to be really kind of wary of immediate acceptance of every single report of these types of biomolecules in dinosaurs. Again, I don't have any doubt that there are biomolecules that are actually preserved in ancient fossils. I have no issue with that at all. But it doesn't mean that I just immediately accept every single report I've heard. All right, that's it. I'm going to quit there. That's hemoglobin challenge number 5. ICR fails again. Brian Thomas, who's the one talking in this video, has recently got a PhD. And what did he study? Soft tissue preservation and biochemistry. And I'm sure that I've been fumbling over my biochemistry language and how to use and what to call these different molecules. I'm not a biochemist, and that's pretty evident. But it's not hard for me to understand the basic literature and to understand that hemoglobin has not been found in these fossils. And it's inappropriate to use the word hemoglobin. You need to use the word hemoglobin remains or breakdown products of hemoglobin have been found. And that provides evidence that hemoglobin was once present in that particular location or in that particular bone, which is not shocking because they had blood in them at one time. And so we find actual evidence that blood was there. But what's there is not fresh blood. It's not fresh hemoglobin. These are not fresh molecules that are the same as the molecules in the actual living organism. They have experienced a lot of change. and they have reached a point where they have degraded, and then they've reached a point where they've become very stable. And once stabilized and under the conditions that they are preserved, they remain stable for 100 million years. And this is not shocking nor surprising. I have another paper that I was going to talk about with the hemoglobin challenge at some point, but it has to do with meteorites and organic molecules that are found on meteorites, right? And so, and actual materials brought back from space have organic molecules in them. All right, so don't tell me that organic molecules somehow just have to break down in a few hundred years just because they're organic molecules. There are organic molecules that can survive in very intense situations. And that's what we're seeing here. We have organic molecules that are capable of surviving for 100 million years. Or in the case of these meteorites, organic molecules that are potentially 1 billion years old. All right, so that's it for me today. Let's wrap it up there with hemoglobin challenge number 5. I have the feeling there's going to be plenty of opportunities to talk about this again, and we'll take a little quick look at some other interesting papers with the future misuses by young Earth creationists of language surrounding soft tissue analysis. All right, that's it. Thanks a lot. Bye-bye.